This is a production of Cornell University.
So once again, thank you, Gaurav, for the invitation. And I'll talk to you about secretory structures. Not everyone may have heard about those, but I hope to convince you that they are actually important. And diversity is really a theme. It's an opportunity, but it's also a curse. So sometimes we're spread too thinly, and you will probably recognize that as well. So let me see. How do we go? Let's just use these. Okay. So I want to put out a hypothesis. This is not entirely proven and tested, but I think it's an interesting one to start for a talk. Chemical diversity of metabolite correlates with the availability of suitable storage. So in a way, when you look at plants, they make hundreds of thousands of different metabolites across the different species and genera. Humans can't do that. And one of the reasons is because plants, I think, have evolved these secretory structures where they can use them as a playground for testing out new biochemistry. You make something new, it's not immediately detrimental to growth because you can sequester it. And that's a key aspect of this. So that's the hypothesis that I'll go by. Now, the other possibility, obviously, is to volatilize metabolites if you can get rid of them by moving them into the atmosphere. But that's not something we'll focus on today. So you can look at the evolution of land plants in various different ways. You can look at the fossil record. You can look at extant plants and try to make inferences as to what things may have looked like a long time ago. You can look at biomarker metabolites that are indicative of certain metabolic capabilities. You can look at structures that exist on plants today. And when you do that, you notice that there are various different structures that have evolved over time. One important one was the evolution of oil bodies. So if you look at the non-vascular plants, you have hornworts, you have mosses. They're not very rich in chemistry, but the liverworts are incredibly rich. And they have oil bodies. They are intracellular organelles that hold lipophilic metabolites. And at least from the correlative evidence, that might be one of the reasons why they can generate this diversity of structures, because they can sequester them. Then what people have also, boy, this is difficult. What people have also shown is that there are ambers that people have recovered from more than 300 million, that have been dated back to more than 300 million years ago. So those are fossilized remains of what very likely were resin ducts or resin blisters. You still find them in the extant conifers today. And again, it's an important storage structure. Then you will find glandular trichomes. Now these were, the earliest ones were found at a similar time, dated back to the transition of the Carboniferous to the Permian period. And it was seed ferns that had these glandular trichomes with hooks on top. So if an insect would walk over the surface, it would catch this hook, and that would then release volatiles very likely that were stored in those glandular trichomes. Now they're very common in the angiosperms, which evolved much later. But they were also, it looks like, present in some of the plants that were present on Earth much earlier than that. So you will find these various different themes when it comes to these secretory structures. Now I want to make a pitch very briefly about studying these metabolites that accumulate in these specialized structures. And that is that this is not what people would generally consider as metabolomics. It's one of these buzzwords that people like to use these days because there are lots of considerations that have to go into your experimental design. And I just wanted to go over this very, very briefly. So if you're looking at, oops, let's go back. If you're looking at volatiles, in some cases you can collect them from the headspace, but you have to be equipped to do so. You can do a solvent extraction. What's become very popular is tissue imaging. and I'll show you a few pictures of what that looks like and what you can learn from those kinds of technologies. You can, before you analyze, you can pre-process your samples. If you're only interested in a subset of metabolites, you can absorb them to certain types of materials. If as volatiles, you can do solid phase extraction. That's a fairly common method for pre-treatment. Then in most cases, you want to separate them. You have thousands of different metabolites in an extract. And if they all go into your detector at the same time, you might overwhelm that detector. So a chromatographic separation is desirable. You can use stationary phases, as they're called, or different columns that have different polarities. So in that case, you retain the analytes differentially. That separates them. Something that we do a lot because we work with volatiles that are used in the fragrance and flavor industries. We do a lot of chiral separations. Minus limonene smells like turpentine almost, plus limonene smells like citrus. So we are able to differentiate enantiomers, and so are our animals. So it's very important to focus on those chiral separations to ensure that you know which of the isomers you're looking at. And then for larger scale separations, we do the typical large columns that you've seen in chemistry labs. Then you have different ways in which you can detect metabolites. Mass spectrometry is certainly one important one these days. Diode array also gives you information about that allows you to infer to structure. Flame ionization detection is something that you do with GC. But it's very nice for routine screening. It essentially just burns carbon, gives you a signal. But it's non-selective, which is an advantage. Then you can characterize your analytes by fragmenting them. And there are certain rules in chemistry that allow you to infer structure from these fragments. You can do the same with nuclear magnetic resonance, which is even more informative toward structural characterization. And then you still run into that major problem of peak annotation. So you have lots of spectral features that are reported by your instrument, and then you have to use databases to match those with known spectra or spectra of known metabolites, or you develop technologies like Gaurav and his lab do, where you're trying to come up with rules that will allow you to use theoretical databases, essentially. But the take-home message here is I review a lot of papers, and people oftentimes just believe what the instrument tells them. And you certainly can't do that. So just because your top library hit is X, Y, and Z doesn't really mean anything. Oftentimes you really have to dig deeper and do a better job with the annotation. And then what's also important is that you're not just using one technology. You need to look at things from various different angles, separate metabolites using various different techniques, and then also ideally use different techniques for detection. So just a little pitch here. Let's get into some biology. So I talked about these specialized cell types or secretory structures. I'll begin with the Chinese medicinal plants that we've worked with, the thunder god vine, great name, of course. And it's used in traditional medicines throughout Southeast Asia since the 15th century. It's usually the roots that people harvest, and obviously that's non-sustainable. Once you kill the root, you kill the plant. So you have to regrow these plants. They have to be at a certain age before you can harvest them. And you saw those roots before. They were pretty thick. So those are plants that are 6, 7, 8 years old. So it's a fairly slow cycle in which you grow them. The drug candidate that has been pushed the furthest is called is triptolide, which is a diterpene. I'll show you a structure in just a minute. And there have been clinical trials that have been reasonably successful, but there is a sourcing problem because you're extracting from these roots and the yields are extremely low. The reason is because the dry weight content of those diterpenoids is 0.008% of the dry biomass. So you can imagine you're going through enormous amounts of tissue before you get anything useful. Now, there is some preclinical data for some of the triterpenes as well, and I'll briefly mention some of the work that we've done on those as well. But what's important is that the traditional methods that people have been using are great for preliminary data, but it's very difficult to do this at a clinical scale and then at a commercial scale. It's somewhat similar to the situation with Taxol, which some of you might be aware of. It's something that is now produced in plant cell cultures. So Justin Fischetic was a postdoc who now works in the cannabis industry, like almost all the students and postdocs that come out of my lab these days, because those states that have legalized cannabis now require the measurement of terpenes. And while everyone has focused on the cannabinoids, no one knows how to analyze terpenes. So it's hard for me to get students to actually finish, which is good for them.
Could you define higher education?
I won't answer that, but thank you. So anyways, this on the left side here, that is triptolide, a very highly functionalized diterpene. Sinastrol here is a triterpene. Both accumulate in the roots to the highest level. Triptolide you also find elsewhere, but as you can see, this is a logarithmic scale here. So the gray are the sinastrol concentrations. They're very, very high, and some of the different root types that you see here, much higher than what the diterpenes. So yes, the boss sometimes goes into the lab and actually does stuff, particularly when it thinks that no one else likes, like running larger columns and isolating metabolites and those kinds of things. So I did this 3 years ago, I think, over Christmas break. I isolated some of those peaks, and Nayanan Shrividya, or Shri as we call her, who is a senior scientist in the group, did most of the downstream analysis. So she ran them over mass spec. She got NMR spectra, and we developed a database for NMR spectra that has been very, very useful for us in deep replicating extracts. So we can very quickly identify which of the peaks that we find in a chromatogram and then where we have spectra have actually been reported before. And as you can see, I hope you can see that, I hope, that there are a number of these sesquiterpene alkaloids of fairly complex structure, where in some cases they only differ by positions of certain functional groups, or you have functional groups in different locations, and that is very, very difficult to resolve. But even with fairly crude extracts, using this NMR database, we were able to identify or associate tentative annotations that wiggled it down to essentially a structural class. Now, to get full spectral annotation, you would have to do two-dimensional experiments and those kinds of things. So that's the next thing that we want to try, to integrate two-dimensional spectra into the database and find a way to generate an algorithm that will capture that. So anyways, the point here is that these databases allow us to go through extracts much faster than before and focus on the metabolites of interest. And what I'll talk about now are the diterpenes, so the dimethyl sinastrol and the sinastrol that I showed before. So we did root imaging. So in this case, it's a MALDI-based platform. You have a cryo-sectioned sample. You have a laser that goes over that sample. The sample can be moved in a certain pattern, and essentially, eventually you will have covered the entire surface of that sample. On the other end then is a mass spectrometer. So essentially at every given time when you've collected a sample and you know what pattern you use to go across that sample, you get a mass spectrum on the other end. You can then analyze those mass It's an instrument that also allows you to do separations in the gas phase called ion mobility. So the molecules are separated by shape because you can't do chromatography. That's very important here because a lot of these are isobars. They have the same mass, but the shape is different, so you can distinguish them using ion mobility. And as you can see, you have these triterpenes all on the outside where the cortex is, whereas some of these sesquiterpene alkaloids, they are in the cortex. My bad, and the triterpenes are all in the periderm. The nice thing in this case is that the triterpenoids of interest, they're quinone methides, so they have a color. They have chromophores in there, so we can actually see them, and we can kind of correlate what we see with mass spectrometry with what we actually see with the naked eye. But the nice thing here is you can, as the technology improves, you can get down to almost cellular resolution with these technologies. And that is, I think, something that in the future is going to make a big difference. However, and this is the drawback, it's not very sensitive. So for the diterpenoids, for example, we couldn't see them. The concentrations of those were too low to be picked up using this approach. They were somewhere in the fairly high background noise. One of the things you have to do is you have to coat these samples with something that facilitates the ionization. Usually in an ion source, you have a lot of energy to break up molecules or ionize them. With the MALDI, you don't have that. So you need that matrix to help you with transferring energy from the laser all the way to the molecules. But the matrix gives you a lot of background. So that's certainly one disadvantage. So we had to, well, let's stay with those triterpenes. There's one more slide here. So this is another cross-section in this case of a fixed root. And you can see this orangish color here. Now, because they've been fixed and you use solvents to do that, the triterpenoids don't stay exactly in the location where they were before. Because what we were asking is it maybe incorporation into membranes? There are sterols in membranes, like beta-sitosterol would be a common one. So the question would be, is there more of these triterpenes found in the layer where we actually find the accumulation based on the mass spec results compared to other parts of the root? And you see that the outer root parts, if you just if you just section this out, had a bit more beta-sitosterol, which is a typical membrane sterol, than the inner parts of the root. But you have to keep in mind that large parts of the inner root are dead. There's nothing going on, so there are no functional membranes and so on. So the difference is really not that large, whereas for the triterpenoids, the difference is enormous, from high milligrams per gram quantities in the periderm to essentially nothing in here. So we felt that the membrane was apparently not the best candidate as a location. So that got us thinking, and we did a number of different stains for different cell wall polymers. One of them is suberin. And if you look at the localization, then the only one that appears to correlate with the localization of the triterpenoids is suberin. It's a polymer, and there are, in some cases, people have actually reported triterpenoids to be incorporated, but these are soluble. So we can just use an organic solvent and they'll come out. So they're not polymerized into the polymer. They're just somewhere out there, in that general cell wall area, but we don't know exactly where they are. But at least the imaging gave us some good ideas of where to look for. So that brings us back to what did we do with those diterpenes that we couldn't profile. We needed a different system to work on where the concentrations that we would work with would be higher. So Michael Hartmann, who's now at the University of Düsseldorf, was a postdoc, and Amber Parrish, who's still a technician in the lab. They developed a root culture system. These are adventitious root cultures. You can see, you can grow them in fairly large flasks. They grow to kilos weight balls of roots within a couple of weeks. So it's a very nice system. And if you look at the profile here, so those are the roots and metabolites extracted from there. Don't worry too much about the various different metabolites there. Triptolite is here. So triptolite and other diterpenes are secreted into the medium. Doesn't get any better than that. You don't even have to extract from the roots. It's just being placed where you like it. You replace the media anyways. So every 2 weeks, you would take the media, extract them, and those root cultures would keep going. And the concentrations in there would actually be fairly high. So that allowed us to just look at the profile of diterpenes in those root cultures. Again, it was using that same approach where we're combining the analysis by mass spectrometry, MS-MS spectra, and NMR to get IDs on those. And if you look at the structures, you can place them into a theoretical pathway. So this is what we could come up with. You have the universal precursor for most of the diterpenes, geranyl diphosphate. You can make an intermediate, eventually dehydroebiatic acid. From there, you functionalize, so you have to lose this as CO2. You have to functionalize this position here. That would give you an additional hydroxylation here would give you tryptophan B. We did not detect tryptophan B in our cell cultures, but we detected something that's very closely related. It's called tryptophan A, so it's methylated here. You have a methyl, you can cyclize here, that methyl ester, but you can also cyclize at this stage, and both of those metabolites we found in our extracts, and so on. So in some cases, you can just look at the metabolites that you're getting out of an extract like this, and you can come up with a hypothesis of what the biosynthetic pathway looks like, and then you can start trying to work this out, which one of the PhD students, Fima Inabui, did. So she looked at the diterpene synthases. There are quite a few of them. I hope you can see this. So that it's a two-step conversion here to a precursor, a different precursor, and then you have various different terpene synthases that take it to those different structural types. And what was rewarding in this case is that the end products of those generated by those terpene synthases have the structural cores that people have reported to occur in the genus Triptorigium. So they are natural products. These are caurane diterpenes that are not directly related to gibberellic acid. They have the same core structure, but they're actually natural products and not hormones. They don't have more hormone functions. And so those had been characterized before, and we essentially identified the first genes and encoded enzymes on the path to those natural products. The AB18 diterpenes, those are the precursors of triptolite and the other diterpenes in Triptorigium. So that was significant progress there. There are now a number of other groups that are working on this. So because I don't have a lot of funding for it, we're currently not taking this any further. So that allows me to transition to a different project, a different type of secretory structure. Now we'll be talking about glandular trichomes. And one of the plants that we've worked with for many years is peppermint and other mints. And the question was, so we had made significant progress in working out the pathways that lead to the essential oils that you're all familiar with. But what was not known is how they power metabolism, because the glandular trichomes in there are non-photosynthetic. So what are these structures? They are epidermal outgrowth. You can't see them with the naked eye. In cannabis you can, but in most plants you can't. They usually have a series of cells that are highly active biosynthetically for the production of specific specialized metabolites. There's usually some kind of storage cavity we found that this is actually pre-formed, and then essentially that space in between just fills up. And some of the metabolites that you will find in different glandular trichome types would be menthol and other monoterpenes. And peppermint, artemisinin would be a nice example. In sweet wormwood, used as an antimalarial. Savinorine A is the only psychoactive terpene known so far. And then tetrahydrocannabinol, which I'm sure you're all familiar with, just on paper, of course. One of the important questions for us was where does this complex oil composition in peppermint come from? And that involved trying to find out what the different contributions from potential ancestral species were to the very complex hexaploid genome of Mentha piperita or peppermint. And it turns out that two of the grandparents, if you will, were Mentha suaveolens or apple mint and Mentha longifolia or horse mint. Those are diploid species. They occur naturally. They are particularly prominent in the Mediterranean, and very likely that's their origin. My current hypothesis is that it was the Romans that brought a spearmint-like plant over to Britain, where eventually Mentha piperita emerged. Peppermint was first found in a place called Mitchin. It's relatively close to present-day London. Very likely humans did not have a hand in this. It was a chance discovery. Someone went into a field, said, well, this smells nice. And because it's sterile, because of that very, very complex genome composition, 4 copies from Mentha aquatica, which is water mint, very common in England. Then 1 copy from Mentha suaveolens and 1 copy from Mentha longifolia. You can imagine what that does during meiosis. Only bad things. So it's sterile. And that's why everyone's been growing just vegetatively propagated stuff. There's no breeding. There's also no breeding with the most widely grown spearmint, which is native spearmint. It's a triploid. Same story. You just can't do a whole lot with it. But what we wanted to know is how you power metabolism in the cells that actually make those essential oils. I should probably go back briefly. So the contribution from the water mint is primarily a molecule called menthafuran. Then from the Mentha spicata comes, it has a different hydroxylation pattern from peppermint. So this is the position C6, and all the C6 related metabolites tend to accumulate in spearmint. You have a different position that is functionalized in peppermint. It's the C3 position, and you can see that all the products that accumulate in the oil from this pathway are functionalized in that position. So that kind of differentiates the two. So we wanted to know how you power metabolism in there. Sean Johnson, who's now a postdoc with Björn Hamburger at MSU, was very interested in building genome scale models. So in that case, what you do is you summarize all the information you can get on a species of interest. And you put this on top of something that's already known. So the first genome scale models, as you might imagine, were developed for Arabidopsis. They contained a few thousand metabolic reactions, and they were all linked. The key there is that the only parameter that's really important there is mass balance. And then the second one is the stoichiometry of all the reactions has to add up. Those are the only constraints there. So there's no kinetics in there. The only information you need is which enzymes use which substrate, make which product, and what are the cofactors. And all of this across hundreds or thousands of reactions has to add up, which is not trivial. So there's a lot of curation that has to go into building these models and get them to work. So you extract information about those reactions from various different databases. You use as much information as you can that is already existing. Balancing the equations is essentially what I just told you about. You need subcellular localization. because if you have different isozymes doing different things in different compartments, you want to capture that. That then means that metabolites have to be able to move between compartments. So all of this has to be accounted for, otherwise your model dies. And then what we also wanted to do is associate reactions with transcripts, primarily to figure out which of the reactions in this very large network are actually occurring. So if you have pathways where you need 10 reactions and the genes for all those 10 enzymes are not present, well, very likely that pathway is not used in that cell type, which is the definition of a specialized cell type. They don't do a whole lot of things. So you want to know, what do they do? So that's something that we integrated, and there are some algorithms that also allow you to integrate those for flux predictions. But you got to be careful because transcription doesn't really translate directly to enzyme activity. So you have to use algorithms that are based on weighted measurements. So the impact of a change in transcription level will not be as high as it would be if you just if you didn't weight it. So it's still more than, like a presence-absence call. Then you expand your network by the reactions that are known to occur in your plant of interest or your cell type of interest. And you essentially do a few other things that you don't have to worry about too much. And it breaks it down into a system that's a bit more simple. Okay, so you have leaves of peppermint. They have these glandular trichomes. You have the secretory cells within those glandular trichomes. It's a disk of 8 cells. And you import a carbon source. These are non-photosynthetics, so they can't just take CO2 from the atmosphere. You go through a network of reactions and you make monoterpenes. We can measure a lot of the metabolites that are required to make monoterpenes. We can measure all the monoterpenes and sesame terpenes and also the flavonoids that accumulate in the oil. So that gives us a system where we can now predict what is the most likely path through all the different possible reactions toward these end products. And one of the first things that Sean did was in silico knockouts. The question that you're asking in that case is, what's essential? If I knock out a reaction, does that network find some way around and still produce the end product? So you can imagine in central carbon metabolism, there's usually some way around a problem. But once you get into the end product pathways, there's none, which is not unexpected. So essentially all the genes that are directly involved in making monoterpenes and secreting those is lethal. If you modeled this as an in silico knockout, obviously you need to exchange all the essential carbon, oxygen, and hydrogen, otherwise you can't make anything. You need to import sucrose or some other oligosaccharide as a carbon source, but then you also need to worry about energy equivalence. You also need to worry about the electrons for reduction reactions, because in this case it was ferredoxin NADP reductase, or FNR, that was returned as an essential enzyme for producing monoterpenes. Now you might not think as that of being directly related, so that's why we looked into this. First of all, the bioenergetic constraints here. What this shows is essentially relative expression levels based on RNA-seq data for isolated glandular trichome cells. So you can see all the constituents of oxidative phosphorylation and its relationship with the TCA cycle. Everything's expressed. They're there. They're active. And then we asked, well, how does this work? How much CO2 are you producing? How many or what's the quantity of monoterpenes produced from sucrose as a carbon source? So in this case, it's radio-labeled sucrose. We tested various different concentrations, and then we used oligomycin, which is an inhibitor of the ATP synthase. So in the presence of oligomycin, you should not be able to generate ATP anymore if this is the process by which you're making ATP. And then at very low sucrose concentrations, you just don't make a whole lot of monoterpenes. Well, that makes sense because if you don't have carbon, you can't make anything. You go a little higher. This is 1 millimolar in, and these are experiments with isolated secretory cells, I should mention. That's weird, you know. So these are the controls, and you're making less monoterpenes, but you're still making them. And if you go to higher sucrose concentrations, there's really no difference. So there needs to be some other process that also provides energy for these cells that is not related to what I'm showing you on this slide. So one possibility, it's not very common to talk about fermentation in plants, but they actually do this. Pollen tubes, when they grow, rely on fermentation because they grow pretty fast. These cells have to make terpenes very, very fast. In peppermint, the oil filling process of an individual trichome takes only 25 to 30 hours. It's an almost visible quantity of oil produced by 8 cells in 24, 25 to 30 hours. Enormous flux. So maybe it's fermentation. So what you do is you're coupling the recycling of NAD and NADH to the production of ATP at the bottom end of glycolysis. You're generating ethanol in this case, and that's a possibility. Microbes do this all the time, and they do it well. So again, the modeling can help you a little bit because you can ask, if I allow certain imports and if I allow certain exports, would you predict any role for fermentation if I allowed these reactions to be present? So you can model without, you can model with, you can have various iterations of this. And at least our initial calculations indicated if you could do fermentation, you would actually get a fairly high carbon flux. It would benefit the entire system. So we then went back. Sean did a number of stainings. You can stain for alcohol dehydrogenase, which is one of the key enzymes to make ethanol. This is on the plant, so you can see that some of the glandular trichomes start to stain very darkly. In this case, now not all of them stain, because the ones that you would stain are the ones that are actively secreting. And there's a developmental program that leaves go through, and not all of their glandular trichomes are active at the same time. So it depends on where you are on the plant and at what stage you are, if you're actually actively secreting. With the isolated secretory cells, you can see that there's different levels of activity, but there's certainly activity there. We then also looked at trichomes that were isolated from young leaves, where you have a higher proportion of glands in secretory phase, with those of mature, and the labeling went from more than 10% of all the glands being labeled to essentially 0. ADH activity was very high in young glandular trichomes, was essentially lacking in mature ones. We had 2 isoforms that were detectable in our transcriptome data sets. We expressed them, purified them, characterized them, and they had the typical Km values of alcohol dehydrogenase, which are actually pretty high. And the Vmax values that are also fairly consistent with what's in the literature. So there might be an alcohol dehydrogenase. We might actually be going that path, but how does that relate to the data that I showed you before? Now, if the Km for the substrate of the alcohol dehydrogenase is very high, it means you need pretty high concentrations before this becomes a viable source of recycling NAD and NADH and producing ATP. And that's consistent with, you need a whole lot of sucrose to get enough flux into fermentation before it actually makes sense. And we estimate that the sucrose concentration, unfortunately, you can't measure sucrose in those cells because as you isolate them, they empty out all the small molecules. And then you have to refeed from a carbon source, which is an advantage in some ways, but you can't determine how much is in those cells unless you had a sensor or something that would give you in vivo concentrations. But at least as a model, it makes sense that fermentation would support the production of those monoterpenes. And again, I apologize. I like these very complex schemes with lots of stuff on there. So this is the pathway from precursors all the way to lots of various different monoterpenes. It's essentially the same pathway that I showed you with structures before, but here it's the genes and enzymes that have been added. I just wanted to focus a little bit on those redox active enzymes, which I've blown up here. So I told you about the ferredoxin NADP reductase, or FNR. Now, in non-photosynthetic cells, the transfer of electrons works in the opposite direction as in photosynthetic cells. So this has been established in roots. The redox potential of the enzymes are different in that you're actually starting with NADPH. You're transferring a hydride, which translates essentially in the transfer of an electron. But you're going from NADPH to FNR to re-reducing ferredoxin, which is the active enzyme that interacts with those reductive enzymes in the pathway toward monoterpenes. So this is the precursor pathway that provides the precursors for monoterpenes. So you actually do need this enzyme in this process. We had never really thought about it that much, but the modeling essentially pointed us toward the FNR as a potentially important player in this. So that's one way you can use these models. So what Sean then did is he looked at all the different isoforms. And again, I hope you can see this. So these are the isoforms of FNR that occur in glandular trichomes and roots. And they all cluster together across various different species. Then you have the photosynthetic ones, which are different redox potential, different sequence, and so on. And again, they kind of cluster together. So that all makes sense. The surprise was here that when we looked at the ferredoxins, so the substrate for the ferredoxin reductase, you still have the photosynthetic ones. They cluster together. You do have a minor, very lowly expressed isoforms in glandular trichomes, and we did this with glandular trichomes from various different species, and they cluster with the root isoforms that have been characterized. But you have a separate one, which is the major isoform in all non-photosynthetic glandular trichomes, in the mint family, for example, and they cluster separately. So it appears that there might have been an event that led to the evolution of a separate clade of ferredoxin enzymes that is present in glandular trichomes. So how does that all fit together? The major and minor isoform, if you cut off the placidial targeting sequence and look at what we believe is the mature protein, there's 12 differences in the sequence at the amino acid level. So you have 12 possibilities of affecting the interaction of the biosynthetic enzyme, the electron transferring enzyme, and the source of electrons, which is the FNR that you need to re-reduce the ferredoxin. We don't think it's a difference in redox potential because that wouldn't make a whole lot of sense. We believe it's actually a difference in the interaction between the proteins. So we've been able to do a little bit of modeling, but obviously you need to test that. And that is something that Nate Boyer is now beginning to do, a graduate student. So here you see the pairwise interactions between, in this case, the FNR and ferredoxin. And there are two residues at the interface where electrons are being transferred that could be important. And this is the interaction between the ferredoxin again and the HDR, so the biosynthetic enzyme involved in monoterpene biosynthesis. And there is a total of 6 residues that are in locations where you could, where an interaction might occur. So it becomes, if you can measure the heats that would be differential during the interaction of those different enzymes, then you can determine if the protein-protein interactions have an effect on total flux through that enzyme. So that's something that we're trying to do now. Obviously, you need super clean proteins for this. You have to go to absolute homogeneity. And then you can use isothermal calorimetry and other methods to really study the interaction. So that's something that we're trying to do, but we're not yet there. Then we asked, well, is this something that is transferable? So can we transfer what we've learned with mint to other glandular trichome-bearing plants? Because quite a few of them, including tomato, that a number of people here are interested in, are photosynthetic. So does what we've learned about mint apply? So we did genome scale modeling for all the species where glandular trichome specific transcriptomes were available. Whoops, I'm sorry. Now, in this case, we only used Illumina data sets because it's very difficult to get some of these algorithms to work if you take data from various different sources. So that's why tomato is missing. Tomato is, the stuff that's been released on glandular trichomes is old enough that it's still ESTs and not Illumina. So we couldn't find data sets to use. So we couldn't include tomato. But there's other species that you'll see are interesting. So here's the grand scheme. You have some that, there it is, that can do photosynthesis. So you can generate reducing equivalent NADP through photosynthesis. You can couple this to the incorporation of CO2, like you do in photosynthetic cells. You generate various different end products from various different intermediates depending on the type of trichome you're looking at. Usually, they will be specialized, for example, to accumulate terpenes, certain types of terpenes. Maybe they make small amounts of flavonoids as well. There will be others that make large amounts of flavonoids, but maybe not something else. There will be some that accumulate acyl sugars, but maybe not too many of the others. Bitter acids would be in hops, and so on. So you wouldn't have all of these reactions present in all of the trichomes, but you can capture the diversity in metabolic pathways using these metabolic models. So what we were asking is, first, if you just look at gene expression patterns, do they match what we would expect? Does the abundance of transcript somehow correlate with the abundance of end products? And the answer is yes. So for salpiglossis, which accumulates acyl sugars, but primarily flavonoids, flavonoid biosynthesis genes are highly expressed. You see some acyl sugars, but it's not a whole lot. Then Solanum clituense accumulates more acyl sugars, less flavonoids, but still decent quantities. And you can see that's reflected in the genes that are known to be involved in those processes. This is a salvia. It makes primarily diterpenes, and all the pathways that are involved in diterpene biosynthesis are highly expressed. Same for Salvia divinorum, which makes a different set of diterpenes. Then Mentha piperita is very rich in monoterpenes, has some sesame terpenes, and again, that's reflected in the gene expression patterns. Bitter acid biosynthesis in hops is just huge. The lupulin glands, there's almost nothing else in there. Pretty amazing. Then you have alkaloids in Camplotheca, for example. Sesquiterpene lactone in something called xanthium. And then Arabidopsis that doesn't make anything in particular. So you don't find those transcripts as being enriched at all. Now, the other thing that was interesting, if you look at the size of these bubbles there are two there are two species here that are known to be non-photosynthetic, where the glands are non-photosynthetic, and that's hops and peppermint. And in both cases, the number of transcripts, the relative number of transcripts devoted to making these particular end products is the highest. So the degree of specialization appears to be even higher in those that are non-photosynthetic. And it might just reflect the fact that if you're partially photosynthetic, you can at least use other pathways to supplement. Here you have to be even more specialized. So I apologize for the complexity of this slide. This is modeling various different possibilities. So different levels of photosynthesis, photosynthesis unrestricted, and sucrose uptake unrestricted, both unrestricted. Here it is reduced sucrose uptake, but still photosynthesis at full power, 50-50, no photosynthesis, I believe, and high sucrose uptake. Those were the four different scenarios that we modeled for all the different species. And I'm just going to pick one out. Once again, it's very difficult to see all the different colors, but what's highlighted here is with the blue squares. Those are reactions that we would usually associate with heterotrophic metabolism. It's breakdown through glycolysis and so on toward a certain end product. And you can see whenever you have sucrose present in high amounts, if you allow sucrose to be used, that's what they will do primarily, even if they're photosynthetic. And there's actually experimental evidence from Alain Tissier's lab in Germany. He worked with tomato, and he actually showed by feeding experiments that is true. So even the photosynthetic trichomes of tomato prefer an imported carbon source to doing their own photosynthesis. So although they're photosynthetic, they're only partially photosynthetic. But if you reduce the amount of sucrose available, then they will resort to photosynthesis if they can. If they can't, then everything breaks down and they don't make anything. So you learn at least a little bit. And I'll race through the rest very quickly. I think we're pretty much out of time. So one of the important questions is obviously, okay, we're beginning to learn how these specialized cell types work, what they do, how they function, but can we extrapolate this so that we can make predictions at a macroscopic level? The question is yes, we can. So we first did this with peppermint. That was Rigoberto Rios, who's now a professor in Colombia. He counted glandular trichomes on leaves at any given stage after leaf emergence, and you can see the numbers are pretty high. So you have to be a special kind of person to do this kind of work. So you have these grids, you can count them, and we've been able to associate the various different enzymes with different development stages and so on. So essentially the models that we've built can now be used to go from a single cell type to the entire leaf, and you can do the same for the entire plant. We've done something similar for resin ducts in loblolly pine. This just summarizes work that took many, many years to do, still unpublished. So anyways, this is a cross-section through a needle, and you can see resin ducts here, and there's a smaller one emerging here. So you can determine the volumes of these resin ducts by microscopic measurements, and you can extrapolate to 3D structures using various different assumptions. And when you do that, we can also measure the resin, the oleoresins that are being produced. So when you project how much resin should be there based on just microscopic measurements and then extrapolation, which is here, to those that are actually measured, we've done this for 3 entire trees, big trees. It is actually fairly close. So if you understand what's happening at the single cell level, you can, in some cases, if you're willing to put in a lot of time and you have very patient people, you can extrapolate to entire organisms. last example where this is South Sea. Oh, I'm sorry. This is South Sea Vu, not Rigo. She became a Buddhist nun after she finished. So I'm not sure if it's for everyone to work on these kinds of projects. She worked with citrus peel. Her model system was grapefruit. And you can see they have different shapes at different developmental stages. So you have to capture this mathematically when you're going through all your thousands of microscopic images. You have developmental patterns. So you have fruit size versus volume per cavity and the distribution of different cavity sizes over fruit development and so on. And you can make predictions of how much oil should be accumulated in the peel. And again, the dotted line is what we predicted. The solid line is what we measured. And this was over 2 growth seasons for entire fruit. So it actually works reasonably well. So you can go from single cell types again to entire fruit. So anyways, I think I've mentioned people who are involved in my lab in the work. For those that might be interested, you can look at our website. We do other things as well. We also have a service center that might be of interest to people. We've done a lot of collaborative work with Ruben Peters' lab on the Triptorigium project. The others were not as collaborative in this particular case. Funding came over the years from lots of different sources for the project, but also for individual people from the NIH, Fulbright, and the Human Frontiers program. And I believe that's the last slide. So thank you for staying with me.
I was fascinated about the fermentation producing ATP. So are you saying it produces ATP at a faster rate, but less efficiently than respiration?
Okay, so the question is, do those cells then produce ATP less efficiently when they use fermentation than what they would do if it was through oxidative phosphorylation? And the problem is it's very difficult to quantify those processes. We've looked at enzyme activities. We've not measured ethanol production, which would be what you would have to do for fermentation. You can label, but it's trying to quantify these exactly when you're working with these isolated cells is not trivial. Then when you're measuring CO2, you always have to take into account that it's a process where you're trying to capture this. And some of the monoterpenes or other volatiles will be captured as you're trying to measure CO2. So we've not found a technical approach that we found was quantitative enough to really answer the question. We would love to know how much contribution you have from the two. So far, it's, they must be doing either one or both, which are not mutually exclusive, but quantifying this has been a real challenge.
It fits a little bit with Otto Warburg's finding that cancer cells use fermentation as opposed to respiration.
Okay, yeah. Yeah, and you would think they do this when, for some reason, oxidative phosphorylation is not at its peak, or they can't use that in the same way they would usually do. So it would almost seem like it's a supplemental thing they would do. Or the carbon source is high, which changes the kinetics, and it just makes it possible to do that. Yes.
Like in power tubes.
Right, yes, exactly.
Time for one more question. So I have in your transcriptome data, when you looked at different species, did you find some specific type of transporters being enriched across all species? It looks like there's some conserved central metabolites that are going into the trichomes repeatedly.
Yeah. So I don't know if everyone could hear this. It's a question about secretion and transport. And it's been something that's bugged us for many, many years. When we did ESD sequencing, which is what you did before some of you were born, then you would look at the abundance of these ESTs, and you would notice that there would be things like lipid transfer proteins. In Peppermint, it was the most abundant transcript in glandular trichomes, more than 1% of all the transcripts present. We did some structure functional modeling at the time. And so lipid transfer proteins are supposed to transport lipids from one membrane to the other. And you can fit, if you look at the it's solution state structures, NMR structures that they have for the lipid transfer proteins, you can fit one fatty acid chain in there. And then the rest just has to kind of sit there and it moves them. For terpenes, they have a different structure. They're much smaller, but they're a little bulkier. And we could not come up with any mechanism that would give you any specificity. So if you try really hard, you can fit a monoterpene in there, but it never felt right. So there are some transporters that people have found, mostly through genetic approaches, but in some cases also biochemically. Natalia Dudareva reported one recently. There's been some older works on roots that secrete diterpenes. So people have found ABC transporters that when you make mutants, then are impaired. There are lipid transfer proteins in tobacco that have been associated, but never at a structure function level shown to actually work. So there's evidence, genetic evidence, that this could work. The biochemical evidence is still somewhat weak, and I'm still worried about specificity. So I don't know how these things would be specific enough for the types of volatile blends that you're producing that would then be secreted. But it's something that everyone wants to figure out, but it's a tough challenge.
This has been a production of Cornell University, on the web at cornell.edu.